Hello students, a very warm welcome to CBC classes. Here we are going to discuss about worksheet 3 questions. Okay, so our very first question over here is question number 1. So, important site for the formation of glycoproteins and glycolipids is first option A, lysosomes, second plastids, Golgi apparatus, and fourth one is vacuoles. So here if you see the formation of glycoprotein and glycolipids, this happens in the Golgi apparatus. Yes. So Golgi apparatus is the correct answer. Golgi bodies you can also call it as. So this Golgi apparatus, basically what is the function of the Golgi apparatus? It packs, it packs and modifies, and it transport substances or that are formed in endoplasmic reticulum to various parts of the cell, and extracellularly. So both intracellularly and extracellularly the transportation of substances is done by Golgi bodies. So the substances that are modified, like proteins and lipids, are modified into glycoproteins and glycolipids. Okay, so these glycoproteins and glycolipids, which are packed, modified and transported to various parts of the cell, that is intracellularly and extracellularly. So here the question answer, important site for the formation of glycoproteins and glycolipid is Golgi apparatus. Now coming to the next question, which one is an organelle within an organelle? So this is an organelle that is present within another organelle. So first suppose this is an organelle, this is present on the organelle or inside the organelle. Okay, so lysosome, ribosomes, mesosomes and endoplasmic reticulum. Lysosomes are single membrane structures which consist of hydrolytic enzymes, right? So this is not the correct answer, just I am highlighting the points. So lysosome is the one which is present in, lysosome is a single membrane structure which consists of hydrolytic enzymes, right? And what about mesosomes? Mesosomes are present in prokaryotic structures which are helpful for respiration. And endoplasmic reticulum, these are flattened structures that are present close to the nucleus, right? They are very close to the nucleus and they are helpful for formation of proteins and lipids. Smooth endoplasmic reticulum form lipids, whereas rough endoplasmic reticulum form proteins. Now organelle, ribosome is the one. Why? Because it is present on rough endoplasmic reticulum. Okay, on rough endoplasmic reticulum, the rough surface is because of ribosomes, and it is also present in mitochondria. Okay, mitochondria, it is present in mitochondria, it is present in chloroplast structure. Okay, so the ribosomes are present in chloroplast. Chloroplast is the organelle, right? And another organelle, endoplasmic reticulum, specifically rough endoplasmic reticulum, consists of ribosomes. Mitochondria also consist of ribosomes. So this is present within another organelle. That is the reason it is called as one organelle present in another, within another organelle. So here the answer is going to be the ribosomes. Okay, now moving to the next question. Smooth endoplasmic reticulum synthesis. The main function of rough and smooth endoplasmic reticulum. Let us see, the rough endoplasmic reticulum is having ribosomes on it, so they are helpful in protein synthesis. Whereas smooth endoplasmic reticulum doesn't have ribosomes and they are involved in lipid formation. Okay, so these are the two types of endoplasmic reticulum over here. Endoplasmic reticulum, abbreviation ER. Okay, they are classified as RER, which means rough endoplasmic reticulum, whereas smooth endoplasmic reticulum. Okay, so these are rough endoplasmic reticulum and smooth endoplasmic reticulum. And what are the major functions of rough endoplasmic reticulum? Protein synthesis, right? And what about smooth endoplasmic reticulum? Lipid synthesis. If you see other functions of smooth endoplasmic reticulum, detoxification, they are present more in liver, so they remove unwanted or toxic substances from the body. Okay, so lipid synthesizing, detoxifying are the main function of the smooth endoplasmic reticulum. Okay, so here in the question, the smooth endoplasmic reticulum synthesize proteins. No, it doesn't synthesize proteins, it doesn't synthesize carbohydrate, it forms lipids. So answer is going to be the lipids. Okay, now moving to the next question, which of the following cell organelle is present in highest number in metabolically active cell? So here you have to highlight a point, highest number of metabolically active cells. What do you understand by metabolically active? The cells which are having high rate of reactions like catabolic or anabolic reactions, then they are called as metabolically active cells. If this is a cell and this is active, metabolically active, which means anabolic activities are happening, catabolic activities. Catabolic releases energy, whereas anabolic takes the energy, right? These kind of reactions like respiration or you can see photosynthesis reaction that is happening in the cell. Okay, so these reactions, digestive reactions. So when the cell is metabolically active, what it needs? It needs energy. So energy is in the form of ATP, adenosine triphosphate. So the cell requires large amount of ATP. Why? To perform various activities, So a metabolically active cell, which means metabolically active cell means ATP is required more in amount. Metabolically active cell, which means it requires energy, high amount of energy is required, and energy is in the form of ATP. And where does ATP is formed? ATP is formed in mitochondria. So mitochondria is called as powerhouse of the cell. That you must have learnt in your earlier classes also, that mitochondria is the one which forms the ATP. That is the reason it is called as powerhouse of the cell. So more mitochondria, more ATP generation and more activity that can a metabolically active cell can perform, right? So if you need more amount of ATP, the more amount of mitochondria should be present. So a cell consists of more amount of mitochondrial cells to perform various kinds of activities. Okay, so the cell will consist of more amount of mitochondria. So here the answer is going to be 1 mitochondria, second Golgi apparatus, endoplasmic reticulum, or 4 lysosomes. The first one mitochondria. Now why Golgi bodies or endoplasmic reticulum or lysosomes are not present in high number? Because they don't produce ATP. Here Golgi apparatus produce glycoproteins and glycolipids. They modify the lipid and protein structure. Now what about endoplasmic reticulum? The endoplasmic reticulum is involved in formation of lipids and protein or biosynthesis of plasma membrane, which means synthesis of plasma membrane, repairing of the tissue. Okay, now coming to the lysosomes, lysosomes removes the unwanted substance from the cells. Okay, so lysosome consists of enzymes which break down the dead or not required parts or damaged organelles from the cell. So these are the specific functions, and mitochondria is the one which forms the ATP. Chloroplast, if you have a chloroplast in the options, chloroplast on the other side, it prepares glucose. It forms glucose from the light and carbon dioxide through light reaction and dark reaction. It prepares the glucose, and that glucose is break down in mitochondria through series of reaction. Glycolysis, link reaction, Krebs cycle, electron transport, chain system. That is what we have learned. Through at last, 38 molecules of ATP is generated from a cell in aerobic respiration, right? Here also we are learning the metabolic actively cell, which means it requires larger amount of mitochondria and larger amount of ATP is generated. Now going to the next slide, question number 5. The osmotic expansion of the plant cells. So osmotic expansion, which means water is traveling inside the cell or outside the cell. So the exchange of water through osmosis. Okay, so this is the what you have to understand. Here they are talking about osmosis, osmotic expansion, which means the water is entering into the cell of a plant cell kept in water. So the plant cell is kept in water. As a result, the water is entering into the, yes, cell. This type of is why the cell is swelling, because the, if this is, if this is a jar of water and a cell is placed, okay, what happens? The water travels inside. Why the water travels inside the cell and the cell swells? Because this is a hypotonic solution. Water is a hypotonic, which means the water concentration is more. The concentration of solute is less. Solute is less. The concentration of solute is less, and concentration of solvent, which is water, is more. As a result, the water enters from higher concentration to lower concentration. Higher concentration of solvent to lower concentration of solvent. As a result, the cell will swell. The cell size increases. Now, where does this water go? This water is taken up by the specific structure that is present in the plant cells, that is vacuole. The vacuoles of the plant cells regulate the osmotic expansion. So whenever the plant cell loses water, it will lose water from the vacuole. So vacuole is basically storage of water, minerals, or certain excretory substances. These are very small organelles that are present in the animal cells. Sometimes animal cells don't have vacuoles, but in plant cells the vacuoles are larger in size. As a result, you must have seen in the diagrams also, plant cell diagram also. Let me erase this. The size of the vacuole is always larger. If suppose this is a plant cell, okay, and it consists of cell wall, cell membrane, and the size of the vacuole is very large and the nucleus size is very small. So this vacuole consists of H2O, minerals, okay. So whenever the plant absorbs or water enters into the cell, okay, the water is stored in the vacuole. More the expansion of the vacuole, more the expansion of the cell, okay. And what about cell wall? It is flexible enough to accommodate the water, okay. So here the osmotic expansion, the plant cell are expanding by intake of the water. It is due to vacuoles which are present in the plant cells. Now moving to the next question. A cell organelle contains hydrolytic enzymes. Exactly, we have learned in our very first question, we have just discussed. The hydrolytic enzymes are proteases. Any other example? Nucleases, you can give nuclease example. Lipase, amylase. All these enzymes are hydrolytic enzymes, which are basically breaking down of various substances. For suppose proteases break down the proteins. They break down proteins into amino acids, right? Nucleases work on the DNA and RNA nucleotides, okay. Now coming to lipases, they break down fatty acids, lipids into fatty acids. Okay, now coming to amylase, which broke down the starch into glucose. These enzymes break down the complex structure into simple structures. So they are called as hydrolytic enzymes. They are acidic in nature, which means the pH will be less or more. Of course, the pH will be less. Okay, the pH will be less than 7, then it is acidic. So hydrolytic enzymes are acidic in nature and they need certain temperature or pH to act upon certain complex structures like proteins, carbohydrates or lipids. Okay, so to break down these structures, hydrolytic enzymes are required. Okay, so cell requires amino acids, cell require lipids, and the protein is converted into amino acids or lipids are converted to fatty acid by hydrolytic enzymes, and these hydrolytic enzymes are present in single membrane organelle, that is lysozyme. Lysozyme is a single membrane. It doesn't have double membrane like chloroplast or mitochondria. It is single membrane and it consists of hydrolytic enzymes which are active in acid, which are generally acidic in nature and which break down the complex structures into simpler structures. So that is the reason lysosomes are called as suicidal bags. Why? And any disruption in the cell or cell becomes old, what happens? These lysosomes burst and the enzymes digest all other organelles or cell membrane, everything is digested by these lysosomes. So they are called as suicidal bugs and they have hydrolytic enzymes. Now moving to the next question, question number 7. Which of the following cell organelle is responsible for extracting energy from carbohydrates to form ATP? So in earlier also we have discussed about the mitochondria is the one where the glucose is converted and converted in the form of ATP. 38 molecules of ATP is formed in aerobic respiration, right? This aerobic respiration partially it occurs in cytoplasm, completely it occurs in mitochondria. Okay, so the breakdown of glucose, which means carbohydrate form is glucose, right? Now this glucose is breakdown in the presence of oxygen in forming 38 molecules of ATP. And where does it is happening? It is happening in mitochondria. It is happening in mitochondria. So in mitochondria the breakdown of glucose happens in presence of oxygen and it results in formation of 38 molecules of ATP. So that is what carbohydrates are converted into form of ATP in mitochondria. Where in chloroplast, if you see, the glucose formation takes place. In plant cells, the chloroplast is a site where carbon dioxide and water and sunlight, that is taken up by the chlorophyll pigments. Okay, what happens? Glucose is formed. So what is the structure of glucose? C6H12O6. So the glucose is formed in the chloroplast. If there is a question, in which organelle the glucose is formed or carbohydrates are formed, that are chloroplast. Chloroplasts are the organelles where glucose is formed. Whereas the organelle which is responsible for the breakdown of glucose, then it is mitochondria. Mitochondria breaks the glucose into ATP, whereas chloroplast forms the glucose. So this is two organelles which work in opposite functions. Okay. Now coming to the next question. Question number 8. The Golgi complex participates in fatty acid breakdown or formation of secretory vesicles or respiration in bacteria or activation of amino acids. So here if you have to understand, it doesn't break down the fatty acids. Okay, the lipids are break down into fatty acids by lysosomes, not done by Golgi bodies. Then coming to the third option, respiration in bacteria. Respiration in bacteria is done by mesosomes, not by Golgi apparatus. So lysosome is responsible for this action and respiration is in bacteria is mesosomes. About activation of amino acids, this is not done by Golgi complex. Okay, the formation of secretory vesicles, they form, these are present. If you see the cell structure, if this is the nucleus, okay, close to the nucleus there is a structure that is endoplasmic reticular, right? Endoplasmic reticular are present, and here smooth endoplasmic reticulum is also present. And now after endoplasmic reticulum there is another structure, that is Golgi bodies, cis phase and trans phase. okay, cis and trans phase. So these Golgi bodies form the secretory vesicles. If you see, these are the secretory vesicles. They consist of various substances like glycoproteins, glycolipids, okay. They transfer these glycoproteins or glycolipids to various parts of the cell. Within the cell, it can transfer the glycoproteins and glycolipids to plasma membrane or a membrane of mitochondria, okay. So that is the function of the Golgi bodies. It forms the secretory vesicles. It is a secretory organelle that is present within the cell. Not only intracellular, they can send these substances to the outside of the cell, that is called as extracellular. Okay, so this is it. Now we are moving to the next question. So here the answer is going to be the Golgi complex particles is involved in formation of secretory vesicles. Now we are moving to the next question, question number 9. It is the match the following. So let us see what are the organelles that are present in the column 1 that we have discussed already. We have discussed the functions of organelles, Golgi apparatus, lysosomes, vacuoles and ribosomes. Now in column 2, synthesis of proteins, trap, waste and excretory products, formation of glycoprotein and glycolipids and digesting biomolecules. So I am going to give 2 seconds or 3 seconds gap, okay, so that you can guess the answer correctly. So what is the function of Golgi apparatus? A, 1, 2, 3, 4. The Golgi apparatus is in formation of glycoprotein and glycolipids. So the answer for the A will be 3. Okay. Now what about B, lysosomes? The lysosome function is digesting biomolecules, right? 4th. Now coming to vacuoles. The vacuoles trap waste and excretory substances. In animal cells, it generally is traps the waste or undigested substances or substance that is not required by the cell. Okay. Now coming to ribosomes. The ribosomes are present on rough endoplasmic reticulum, then they are called as rough endoplasmic reticulum, right? And they are also present freely in, yes, cytoplasm of the cell, and they are mainly involved in protein synthesis. So A 3, B is equal to 4. So A is 3, B is equal to 4 and C is equal to, if you see, 2 and D, yes, hyphen 1. So the answer is going to be the first one. So this is how you are going to attempt to match the following. First see column 1. Whatever column 1 is present, now just see the column 2. Now relate what column 1 and column 2 consist of. This is how you are going to attempt. Okay. Now coming to the next question. Which of the following cell organelle is present in the highest number in secretory cells? So mitochondria, Golgi bodies, endoplasmic reticulum, lysosomes. So in earlier question, there was a similar question to this. The cell organelles that is present more in number in metabolically active cell. In metabolically active cell, ATP is required. More amount of ATP required. So mitochondria was more in number, right? ATP is required more in number. Why? Because this is a metabolically active cell. Just now we have discussed metabolically active cell. Now we are discussing about secretory high cells. The cells which secrete, like sweat gland cells secrete, sebaceous gland secrete, right? Sweat gland secrete sweat, right? Sebaceous gland secrete oils and salivary gland secrete saliva. So secretory cells. The glands consist of secretory cells. The example. Now these secretory cells, which organ, what they do? They secrete secretions. Different secretions. Okay. They secrete different types of secretions. So which organelle is required more in number? Is it mitochondria? No, mitochondria is required when ATP is required more. Endoplasmic reticulum basically forms proteins or lipids. It is not involved in secretory functions. Lysosome break down the particles, complex particles. So here the answer is going to be the Golgi complex. So Golgi complex are the one, or Golgi bodies are the one which pack modify substances that are coming from endoplasmic reticulum, and they send it to different parts of the cell and outside the cell, that is extracellularly, by diffusion process or exocytosis process. So these are the one which consist of secretory vesicles. Through vesicles they are going to send these substances intracellularly or extracellularly. So here the answer is going to be Golgi bodies. The cells, which type of cells? Secretory cells will have more amount of Golgi bodies. Why? Because they have to secrete different types of secretions like saliva or any fluids or sweat, or sebaceous gland secrete oils. So this is the answer. Now from here, now we are going to learn about homework questions. Okay, we are going to discuss briefly about homework questions. So these are the questions that must be attempted by you. So I'm going to give 2 seconds time after reading the question and options, and you have to guess the answer. Then I'm going to explain the answer. Okay, so let us start. The cytoplasm of animal and plant cells is transferred by a network of tiny tubular membranous system that divides the intercellular spaces into 2 compartments: luminal side, the cell membrane, and extraluminal side compartment. Okay, so this here you can see the network of tiny tubular membrane structure. Okay, so here if you see the cell at the center, typically we draw the nucleus, and tubular like structures are basically tiny, and tubular like structures are basically endoplasmic reticulum. Okay, so this is the cytoplasm that is present, is called as luminal inside the membrane. Okay, and what about the cytoplasmic extraluminal, which means the cytoplasm which is present outside these endoplasmic reticulum, it is called as extraluminal cytoplasm compartment. Okay, so this membranous structure which is closely present around the nucleus, so that it can form the proteins or lipids or detoxify the cell or different functions of endoplasmic reticulum. So these are the tiny tubular membranes. Now what about Golgi bodies? They are also tube-like structure, but they are forming disc-shaped structure. So here the answer is going to be endoplasmic reticulum. So next, while explaining the question itself, I have answered. From now onwards, you are going to answer this question. Packing of substance for export from cell occurs in the smooth endoplasmic reticulum, Golgi bodies, and lysosomes or nucleolus. So this answer from your side, smooth endoplasmic reticulum or Golgi bodies, packing of substances. So packing of substances is mainly done by Golgi bodies. The substances that are coming from the nucleus or endoplasmic reticulum, what they do? They modify them. They attach, like in lipids, they attach glycogen part, glycolipids, or in proteins, glycoproteins, okay, and other substances, secretory substances, they form and they pack it very well, okay, in the vesicles. So this is the function of Golgi bodies. So you must have answered the question correctly. Now moving to the next question. Which of the following cell organelles are named after the name of the discoverer? So the answers will be, like options will be endoplasmic reticulum, DNA, Golgi bodies, and ATP. So this organelle is named after its discoverer. Whoever have discovered this organelle has given his name to the organelle. Any guesses? You must be knowing the answer very clearly if you have learned about NCERT. In NCERT it is given clearly. So yes, let me answer this question. This is my turn. That is Golgi bodies. Golgi bodies was discovered by Camillo Golgi. Camillo Golgi. So that's how it got Golgi bodies name. Okay, so Golgi bodies or Golgi apparatus that we call. Okay, so who is the discoverer of Golgi bodies? Camillo Golgi. That is the reason he named these substances that are these organelles that are closely present to the endoplasmic reticulum as Golgi bodies. Now coming to the question number 4. Name the densely stained reticular structure having many stacked, flat, disc-shaped cisternae of 0.5 meters to micrometers to 1.0 micrometers in diameter. So these are also tube-like structures but flat. Okay, they have two. Just let me. Okay, through nucleus, a flat-like structures are present which are, okay, these are endoplasmic reticulum. Closely to the endoplasmic reticulum, we have another structure which have cis phase and which have trans phase. Okay, so this is the cis phase and this is the trans phase, and these are disc shaped curvature smaller than endoplasmic reticulum. They have concave phase and convex phase. So this is the concave phase and this is the convex phase. So this side it is called as cisternae and that is trans phase. Okay, and these cisternae are, the diameter of the cisternae is 0.5 micrometer to 1 micrometer. The cisternae are also present in the endoplasmic reticulum, but the diameter varies over here. Okay, so here this answer is going to be Golgi bodies. The Golgi bodies are the one which have deeply stained and which are present closely to the endoplasmic reticulum, which means they are close to the nucleus also. And what are the functions of the Golgi bodies? Yes, generally packaging and transport and modifying. This is the function of Golgi bodies. Now moving to the next question over here, the question number 5. So this is a little bit lengthy question, but you can answer because we have already learned characteristic features of the Golgi bodies. So just we have to mark whether this statement is true or wrong, false. Okay. So because they are asking which statement is false, just mark the true or false. First, varied number of cisternae are present in a Golgi bodies. Yes, according to the function, the Golgi bodies number cisternae change. Okay. So this is a true answer. Yes, it is true. Now coming to the next, Golgi cisternae are concentrically arranged near the nucleus. They are present closer to the nucleus. Endoplasmic reticulum and Golgi bodies are present closer to the nucleus so that they can modify or transport substances from the nucleus to different parts of the cell or extracellularly. So these structures are concentrically, if you see, this is the one after other. They are not overlapping. They are present concentrically. One layer after other layer after other. This is how they are arranged. So the answer for this is also true. Now coming to the third statement, Golgi bodies show polarity. See, is one proximal or forming one or concave phase near the nucleus. If you see, this is called as concave phase, also called as forming phase. Why? Because secretory vesicles combine to form, fuse to form the cis phase. Okay, and distal, which means this, away from the nucleus, the other side, this side is called as trans phase. So this is the trans phase and this is cis phase. Cis phase is concave and towards the nucleus, whereas trans phase is convex, away from the or distal to the nucleus. So this statement is also true, and this phase is called as maturation phase. Why? Because from here the secretory vesicles modified substances, secretory vesicles with modified substances dispatch to various parts of the cell. So this is also true. The cis and trans phase are interconnected. So here these cis and trans phase are not interconnected. They have a certain diameter, they have thickness. Okay, they are not interconnected. So this is false. Now both phases are similar. No, both phases are not similar. Why? Because if you see, cis phase was concave and trans phase was convex in shape. So this is also false. So I have given you, explained about the statements. Now you can mark the answer. So he is asking about false statement. He is not asking about true statement. He is asking about false statement. The fourth and fifth statements are false. So here the answer is going to be third one. Now moving to the next question. Which of the following is correct sequence? 1. Root of the secretory products. So here you are seeing the secretory products. Okay, the secretory products are formed in the nucleus to from endoplasmic reticulum to Golgi bodies to vesicles to plasma membrane, right? So here you have to see the sequence and you have to mark the answer correctly. So endoplasmic reticulum, vesicles, cis region of the Golgi bodies, trans region of the Golgi bodies, vesicles, plasma membrane, which is correct. Okay, now rough endoplasmic reticulum is forming Golgi bodies is forming, this is the structure origination. They are not asking about secretory, they are asking about secretory product formation, not the structure formation. So this is going to be the wrong one. Okay, so Golgi bodies are basically formed from endoplasmic reticulum and lysosomes are formed from Golgi bodies. So they are not asking about the formation, they are asking about the sequence of secretory products that are moving from one organelle to other organelle. So if something is formed by endoplasmic reticulum, that is carried to the Golgi bodies, cis phase of the Golgi bodies through vesicles. Then from cis phase of the Golgi bodies, the substances are transferred to the trans region of Golgi bodies and they form secretory vesicles. These are secretory vesicles which reach the plasma membrane for exocytosis. They diffuse from the plasma membrane and these substances reach to the other parts. Okay. Now endoplasmic reticulum, vesicles, trans region, no, they always go to the cis region, then to the trans region. Lysosome, endoplasm, no, this is also a wrong statement. So correct statement will be the first one. Endoplasmic reticulum, the substance that are formed in endoplasmic reticulum are carried through vesicles to the cis phase of the Golgi bodies, then to the trans phase, then to the secretory vesicles that are formed from trans phase, then to the plasma membrane. So here the answer is going to be the first one. Now, which of the following is important site for synthesis of glycoprotein and glycolipid? This we have already discussed in the exercise itself, in the worksheet 3 itself, right? So the glycoprotein and glycolipids are formed in Golgi bodies. Question number 8. Which of the following is correct about Golgi bodies? Golgi bodies is structurally and biochemically polarized. The main function, yes it is. If you have to see correct statement, then again we will use the old trick, true or false. This statement is true. Why? Because it consists of, Golgi bodies consist of cis phase and trans phase. This is cis phase, this is trans phase, which means they are polarized. Okay, so the first statement is yes, true. The main function of Golgi bodies is packing of material to be delivered to the intercellular targets or secreted out, produced by RER and SER. Okay, this is also true. They basically deliver substances, they modify substances, that is packaging of material and delivering intercellular targets or extracellular targets. So these substances are formed in endoplasmic reticulum, but they are modified in Golgi bodies. This is also true. The products that travel through Golgi bodies are usually modified as they move from cisternae to other cisternae. This is also true, because if you see, this is cisterna side, they are modified over here, and certain substances are modified again in the cisterna phase. So all these statements are true. Okay, so answer is going to be all of these. So whenever you have long questions, you just have to take one more second to read all the options and answer correctly. Okay, just we have to mark, in this case we just have to mark true or false, and ultimately in the answer we have to find the correct option. The distinctive feature of lysosome is that it has lower pH than cytoplasm, or higher pH than cytoplasm, or reduced hydrolysis activity, or double membrane envelope. So we know the characteristic features of lysosome. It is single membrane. Let us note down the characteristic feature so that you can answer easily. Lysosomes are single membrane. Okay, they consist of hydrolytic enzymes and these enzymes have low pH. Why? Because they are acidic. They work in acidic environment. Okay, the pH of the cytoplasm is somehow near to the 7 or little bit less than 7, which is cytoplasm is generally little bit neutral side. Okay, whereas the pH of the lysosome is acidic. Why? Because it has low pH. Okay, so obviously the answer is going to be the lower pH than the cytoplasm. Higher pH of the cytoplasm is not going to be the answer. Why? Because higher pH, which means basic in nature, lower pH, which means acidic in nature. So lysosomes are acidic in nature. Lysosomes are acidic in nature. So here the answer is going to be first, reduce hydrolyze activity. They have hydrolytic enzymes. Why will they have reduced hydrolyzing activity? They will be more hydrolyzing activity. So here the answer is going to be the first one. Now moving to the next question. Which of the following enzyme is absent in lysosomes? The lysosome consists of lipases, carbohydrases, proteases. All these enzymes are present. Just in the previous slides we have discussed, right? What about polymerase? Polymerase is formation. Okay, whereas formation of new substances from simple particles or simple substances. Okay, but whereas lipase, carbohydrates or protease break down the proteins, carbohydrates break down the carbohydrates, lipases break down the lipids into fatty acids. Okay, whereas lysosomes consist of hydrolytic enzymes. These are the three, but polymerase is not present in the lysosome. It is present in the DNA for the DNA formation or DNA replication. DNA polymerases are present. Okay, so the answer is going to be the polymerase, which is absent in lysosome. So we have discussed questions about cell organelles. Purposefully we have discussed about endoplasmic reticulum, types of endoplasmic reticulum we have discussed in these questions, and we have discussed about Golgi bodies and lysosomes. Okay, so now we are going to discuss about heart. Okay, the structure of heart, that's what we are going to discuss. Now let us discuss questions one after other. So the valve which regulates the flow of oxygenated blood from left ventricle to the aorta. Let me draw the structure of heart and let me show 4 types of valves that are present in the heart. Okay. So this is a rough diagram of heart representing okay, it is always tilted and we have atriums. Okay, we have atriums. This is atrium, left atrium, right atrium. This is ventricles we have. Okay, so this is a 4 compartment structure. This is a rough picture. I'm not drawing a proper picture over here. Okay, we have valves. And what are valves? These are for small flap of tissue which act as a one-way door. Only from this door you can either enter or exit. That's what it can happen. Okay, flap of tissue, one-way door. Let us understand. For better understanding, let us see, it is a one-way door. You can only enter. Okay, you cannot go. You have to get an exit sign, exit door to get out from this room. Okay, so now these are arteries. Left atrium. Okay, right atrium, now left ventricle, and this side is called as right ventricle. Now when blood flows from left ventricle to left atrium to left ventricle, so there is a valve, there is a flap like tissue over here. This is a flap like tissue that I am mentioning. Okay, this flap like tissue is called as valve. Which type of valve? Bicuspid valve. which is also called as mitral valve. So this is called as bicuspid. Only two cuspids are present. That's the reason it is called as bicuspid. So now the oxygenated blood that is entering from left atrium to left ventricle. Now from left ventricle it has to go to supply oxygenated blood to different body parts, including brain, stomach, intestine, legs, hands. Every cell and every tissue needs oxygenated blood. So through which blood vessel it supplies the blood? That is aorta. If you see, aorta has an arch. Okay, so this ventricle and aorta have a aorta and ventricle. So here the left ventricle into the aorta, right? The left ventricle into the aorta. They have a valve-like structure, that is aortic valve. That is aortic valve. Now this aortic valve is semi-lunar in structure. So they are also called as semi-lunar valves. Now blood goes to different body parts and exchange of oxygen takes place. Now the tissue consists of deoxygenated blood. Now this deoxygenated blood comes to the heart through superior vena cava and inferior vena cava into the right atrium. Let me take the blue to emphasize the veins: superior vena cava, inferior vena cava. Now blood is filled in the right atrium. Now blood has to go to the right ventricle. So again there is a valve structures. Okay. Now here we have three cuspids. If you see, we have three cuspids over here. That is the reason it is called as tricuspid valve. So tricuspid valve is generally present between right atrium and right ventricle. Blood is filled in the right ventricle. Now blood has to go to the lungs for exchange of gases. Now it will go to the pulmonary vein. So this is pulmonary vein. The blood enters from right ventricle to the pulmonary vein through another valve. So again there is a valve over here. Okay, this valve is called as pulmonary valve because it is present in pulmonary artery. Sorry, this is a pulmonary artery, not pulmonary vein. Okay, sorry, I have just explained, given a wrong name here. The blood, deoxygenated blood from right ventricle to, yes, pulmonary artery. Okay, then this is having a pulmonary valve, which is also semilunar in structure. Now the blood enters from pulmonary artery to the lungs where exchange takes place. Now the blood becomes oxygenated. Now this oxygenated blood comes to the heart through pulmonary vein. So here this is a pulmonary vein and this is a pulmonary artery. Don't get confused. Earlier I have mentioned a wrong name, that is this structure is pulmonary vein. So through this the deoxygenated blood goes to the other structure. So we have total 4 valves: tricuspid valve, pulmonary valve, then we have bicuspid valve, and we have aortic valve or semilunar valve. In this question, the valve, what does valve do? What is the function of valve? It prevents the backflow. So if blood is entering from atrium to ventricle, the blood cannot enter from ventricle to atrium. It doesn't allow the blood to go to the atrium, backflow. Again the blood is coming from ventricle to aorta. So it is preventing, this valve is preventing entry of blood from aorta to ventricle. Otherwise it will be reverse of circulation, right? So to prevent reverse circulation or backflow circulation, these valves only allow one-way flow. That is from here to here, here to here, then here to here and here to here. So there is only one pathway. Okay, so here the answer is going to be semilunar valve, tricuspid valve or bicuspid valve or mitral valve. Let us read the question again. The valve which regulates the flow of oxygenated blood. The oxygenated blood is always present on the left side, right? From left ventricle to aorta, from left ventricle to aorta, which means we have aortic valve, which is also called as semilunar valve because of the shape. So here the answer is going to be semilunar valve. Now let us move to the next question. So coronary arteries carry blood to the, what happens? We have learned about the heart structure. Okay, this is the heart structure, again rough image, or let me draw neatly, otherwise you will be confused. Okay, okay, the left ventricle is larger in size and left atrium is larger in size. Always. Why? Because they have to supply blood to the different body parts. Okay, now the coronary artery they are talking about, coronary, which means heart. Okay, so the blood that moves in this direction from left atrium to left ventricle to here, arc of aorta is present, arc of aorta is present. This arc of aorta carries the oxygenated blood. The arc of aorta carries oxygenated blood. Now this aorta has to transfer blood to different body parts. It has to carry blood to the brain. Okay, it has to carry blood to the lungs. Then if the blood is carried in towards the lungs, then it is carried through pulmonary artery. If it has to carry blood to the stomach, there are gastric arteries that are present. The blood that has to carry to liver also, and many glands or kidneys. Okay, if the blood is carried through kidneys, that is done by which artery? Renal artery. Okay, hepatic artery. So these are arteries that are present. Okay, likewise heart also requires oxygenated blood. Why heart requires oxygenated blood? Why? Because the heart is continuously working. It is contracting and relaxing. It has to generate lot amount of energy. The muscle cells have to generate energy. So this energy is produced from the mitochondria and the glucose is break down. The glucose has to be broken down in presence of oxygen, right? Then only ATP is generated, which means oxygen is also needed by the cardiac muscles. So the cardiac muscles are getting oxygenated blood from arc of aorta through a blood vessel, that is coronary artery. So this is coronary artery. Okay, okay, so this is the arc of aorta. From here the branches of blood supply reach to different parts of the heart. Okay, from arc of aorta, a branch that is coming to supply oxygenated blood to the heart, that is called as coronary artery. The coronary artery is the one which is coming out of the arc of aorta, then it is supplying oxygenated blood to the heart. Now this oxygenated blood, after exchange of gases, what happens? Now it has deoxygenated blood that is carried by coronary veins. Okay, so here we have coronary artery, again we have coronary veins which carry deoxygenated blood and which combine to superior or inferior vena cava. So this is what happens in coronary arteries. So coronary arteries carry blood to the walls of the heart. Okay, what about lung? Lung, from arc of aorta, pulmonary artery erupts. Okay, the arc of aorta branches into many arteries. One of the artery is pulmonary artery which supply blood supply to the lungs. Okay, right and left lung. And what about kidneys? Kidneys get renal artery, liver get hepatic artery. Okay, so different body parts get different parts of the arteries and oxygenated blood. Now going to the next question, tricuspid valve is present. So in previous question we have discussed that tricuspid valve is present between right atrium and right ventricle. So here the answer is going to be tricuspid valve is present between right atrium and right ventricle, right? So they consist of 3 cuspids, that is the reason it is called as tricuspid. Whereas bicuspid consists of only 2 cuspids. It is present between left atrium and left ventricle. Here the tricuspid is present between right atrium and right ventricle, and it prevents the backflow of deoxygenated blood from right ventricle to right atrium. So here the answer is going to be yes, the second one, right atrium to right ventricle. Now going to the next question. The ventricle is thick walled as compared to the auricles. Always the ventricles are very thick, and in fact left ventricle is very thick. Why? Because, let me show the diagram that I have drawn in previous. Okay, so these ventricles, okay, they are much thicker walled, they have more muscle tissue. Why? Because the left ventricle has to supply, has to pump forcefully, very forcefully, which means more amount of muscle should be present. Okay, if more amount of cardiac muscle will be present, what happens? The cardiac muscle contraction causes forceful exertion of blood, forceful exit of blood to different body parts. So it has to transfer blood to brain, oxygenated blood to brain. It has to transfer blood to toes, at the end of the toes, and limbs. Okay, when it is supplying blood to the brain, which is anti-gravity, which means how forcefully it has to supply so that blood can reach to the brain in opposite direction. The heart is present in the chest cavity and brain is present in the skull, right? It has to supply blood to the opposite direction. For this it requires a larger amount of cardiac muscle so that it can contract, these contract muscles can contract and supply with a greater pressure. Similarly, the right ventricle has to pump blood to the lungs, deoxygenated blood to the lungs. So these are thicker, and even thicker if you see the left part is more thicker as compared to the right ventricle. Okay, so here this is the answer. Okay, because they have to supply blood to different body parts. The ventricle is thick walled as compared to auricle because it has to pump the blood to different body parts, whereas auricles have to pump blood to the ventricles only. They don't have to exert or extra pressure. They just have to put pressure that requires the blood to flow only to the ventricles. Okay. Now coming to the next question, question number 5. The heart muscles are striated involuntary, striated and voluntary, smooth and involuntary, non-striated and involuntary. So we have discussed in muscular chapter also, the muscle is made up of 3 types. Okay, generally the muscle is made up of three types of muscles are present, which means the striated muscles or skeletal muscles, smooth muscles and cardiac muscles. The skeletal muscles are cylindrical, unbranched and voluntary, and they have striations, which means light and dark bands, whereas smooth muscles are spindle shaped or fusiform shaped, uninucleated, non-branched and involuntary action, and they don't have striations. Now coming to the heart muscles, which are cardiac muscles. Heart muscles are cardiac muscles, which are cylindrical in shape. Okay, which are cylindrical in shape. They have branches, but they are involuntary in function. Why? Because we are not controlling this muscle. The heart muscles are controlled by the central nervous system. So they are continuously pumping, they are not under our control, and these muscles are multinucleated as well as they have striations, light and dark bands are present. Okay, because of actin and myosin, we are going to discuss in detail in our later chapters. So they have striations and they are involuntary. So striations are present and involuntary. Striations which like zebra have striations, black and white straps. Similarly here also we have light and dark bands. So this is the striations. So the answer over here is yes, the first one, striations and involuntary. Okay. Now here, 6th question, which is very big in the screen. Let me explain you. You have to see the structure of the heart, A, B, C, D, E, F, G. You must be having the worksheet. Okay, open your worksheets. You can see the diagram very clearly. You just have to label the heart structure. You must have remembered about aorta and pulmonary vein, pulmonary artery, ventricles and yes, valves, auricles or atria. Okay, so let us see those structures. over here. The first one over here, or the B, the arch is formed by aorta. So here the A is forming an arch. The blood vessels which form the arch was arc of aorta. Aorta is the largest blood vessels which is carrying oxygenated blood. So here the A is aorta. Now if you see all the options A over here is aorta only. Okay. Now let us move to the next part, B. So if you see, the B is opening from the right ventricle. The B is opening from the right ventricle, which means it will be, let me check again. Pulmonary artery, which means pulmonary artery. Am I right? A is aorta, B is opening from the right ventricle, which means pulmonary artery. C, it is opening into the left atrium or auricle. C is entering the blood vessel which is entering the left atrium, which is pulmonary vein. Now D, they are asking about the chamber. This chamber they are asking about. Okay. So this chamber is D, which is left auricle. Now coming to the E, which is bicuspid valve, or you can say, you can take options over here. E, they are asking, this is the E1. Okay. So this is picture is little bit difficult to understand. Why? Because it is very small in a smaller page. Okay, if you see carefully, the E's represent this structure. So E over here is tricuspid valve. Now coming to the F, left ventricle. Okay, G, right ventricle. H, inferior vena cava. I, right auricle. And J, so basically they are J referring to semilunar valves. This is the semilunar valve that they are pointing out. Okay, now K, superior vena cava. So here A is aorta, B is pulmonary artery. In which case the pulmonary artery is present? If you say B is pulmonary artery, so you don't have to go for other structures also. With this only you can tell that second is the option. A is aorta, B is pulmonary artery, C is pulmonary vein, D is left auricle. Okay, we have marked it. And E is tricuspid valve, that is present between right atrium and right ventricle. Here the labeling have not been given properly in this question, but if you see very closely, the minute arrow can be seen to the, yes, tricuspid valve. Then left ventricle F, G is right ventricle, H is inferior vena cava, I is right auricle, J is semilunar valves, and K is superior vena cava. Okay. Now moving to the next question. The bicuspid valve is present between, we have discussed this already, right atrium and right ventricle, or right atrium and left ventricle. We have discussed about 4 valves, right? The valve that is present between right atrium and right ventricle is tricuspid valve, and the valve that is present between left atrium and left ventricle is bicuspid valve or mitral valve, which blocks or which stops the backflow of blood from left ventricle to auricle or atrium. So here the answer is going to be the left atrium to left ventricle, which means third is the answer. Let me draw it for you where exactly these are present so that you don't have any confusion. Okay, so this is the right auricle or right atrium. Here is this is the left auricle or left atrium, left ventricle. So here is the bicuspid, 2 flaps are present. As a result, it is called as bicuspid valve. Okay, the blood will flow from this to this direction. When the valves open, the blood will flow to this direction, but it prevents the backflow of the structure prevents the backflow of the blood. Okay, from left ventricle to left auricle. So here the answer is going to be the third one. Now coming to the next question, from the following the one which has a thick wall. The thick walls are present. So here basically aortic arch, precaval, postcaval and portal vein. So you must have understand the terminologies. Precaval, which means vena cava, superior vena cava, and inferior vena cava. This is inferior vena cava. So precaval, which means superior vena cava, blood vessels, vein, and inferior vena cava. Okay, they are also thick walled. They have thin wall, okay, and they are veins. Basically, they carry deoxygenated blood with less pressure. And this is portal vein. These veins are present in liver. Portal, which means hepatic veins or portal, they are present in the liver. Now aortic arch, this aortic arch rise from the left ventricle, and this is thick in walls. Why thick walls? Because it has muscles. These aorta have muscles and elastic fibers. Why it is they are having? Because this aorta has to supply blood throughout the body through anti-gravity also to the brain and spinal cord, to every structure that is present in the body, right? So this arc of aorta is very thick because it is made up of muscles and elastic tissue which transfers blood to different body parts, and these muscles contract very forcefully so that they can send blood to different body parts and to prevent the leakage. And the blood that is flowing in the aorta has high blood pressure, to prevent the rupture of these blood vessels due to this blood pressure. Okay, the arc of aorta is thick. So answer is going to be arc of aorta. Now moving to the next question. The heart is enclosed in a membrane structure. So heart is also like pleura membranes are protecting the lungs. Heart is also protected by certain structure that is called as pericardium. The pericardium is further divided into epicardium, myocardium and endocardium. Okay, so here the heart membrane, mucous membrane, cell membrane are not the structure. The heart is complete heart is enclosed in pericardium. This pericardium prevents from the shock or the stress, external stress or internal stress. Now moving to the next question. The smallest blood vessels in the body is, so the blood vessels, we have three types of blood vessels, that is arteries, veins and capillaries. So arteries and veins are large vessels which are carrying oxygenated and deoxygenated blood respectively, right? Arteries are carrying oxygenated blood, veins are carrying deoxygenated blood. Now what about capillaries? These capillaries are carrying the oxygenated blood to the smallest structures like tissues and cells, because these blood consist of glucose, oxygen, important factors or important necessary nutrients that are required for the cells or tissues. Now from tissues they collect the carbon dioxide, they collect the waste substances. Okay, these capillaries, they collect those substances and they join to form veins. So basically capillaries are fine network of blood vessels between arteries and veins. Okay, so these smallest blood vessels are capillary. Okay, they are very fine network. If you see on the arms also, you can see vein branching. Veins are branching into smaller structures, very small structures that are not visible on the hand. Till some point you can see these branches. After some time these branches are not visible. Okay, they are very thin. So these are the smallest blood vessels, capillaries. Now coming to the next questions, homework questions. So these questions are for you. I'm going to give some time for you so that you can attempt those questions and we can discuss the answers. So let us discuss the function of valves in the heart is to increase pressure, allowing mixing of blood, prevent black flow and regulate heartbeat. From the starting first question of this worksheet, we have discussed that valves prevent the backflow, that is from ventricles to auricles or from blood vessels to ventricles. We have four valves, that is tricuspid valve, bicuspid valve, aortic valve and pulmonary valve. Aortic and pulmonary valve are also called as semi-lunar valves because they are in the shape of moon, crescent moon. Okay, now the answer is going to be prevent backflow. Yes. Now moving to the next question, question number 2. The heartbeat originates from SA node, AV node and Purkinje fibers or bundle of His. So what are the structures you must have learnt in the classroom also? Let me draw the heart structure so that you can understand this better way. Okay. Okay, these are articles or auricles and ventricles. Now if you see, SA node is present. Okay, SA node is present near over here, near where the superior vena cava is coming inside, here exactly the SA node is present. Okay, now SA node, from SA node the impulse is transferred to the AV node. Okay, so what is SA node or AV node is doing? They are forming the heartbeat. How? The electric impulse that is generated in the SA node is transferred throughout the auricles near interseptum of auricles. Okay, now this A node holds the impulse for a second. Why? Because when impulse are traveling from SA node to AV node, the auricles contract. When auricles contract, the blood enters into the ventricles. So when this happens, the valves open. When auricles contract, the valves open and allow the blood flow to the ventricles. Okay, now the ventricles have blood. From here, from AV node, the impulse is transferred to the two branches, that is bundle of His. First the impulse is generated from the SA node. Here this is AV node. The impulse is generated to bundle of His, then to Purkinje fibers. If you see, the fibers are having thin minute structure so that they can transfer the impulse throughout the ventricles. Now bundle of His and Purkinje fiber, what they are doing? They are transferring the impulse. They are transferring the impulse toward the ventricles so that now ventricles can contract and push the blood into respective pulmonary artery and arc of aorta respectively. Okay, so this conduction of impulse is generated from SA node. From SA node to the AV node, what happens when impulse travels from SA node to AV node? The auricles contract. Only auricles are contracting. Now when AV node to bundle His, it takes some time. Why? Because the complete elimination of the blood to the auricles, complete filling of the ventricles should happen before the contraction of ventricles start, right? So here again the bundle of His and Purkinje fibers, what they do? They contract because the bundle of His and Purkinje fiber consist of the impulse. Okay, so here the heartbeat originates from the SA node. That is the reason SA node is called as pacemaker. So answer over the question is SA node. Now let us move to the next question. The pacemaker of the heart. Just now we have discussed the heartbeat starts at SA node. So SA node is called as, yes, pacemaker of the heart. Now let us move to the next question. The heart of a human. Heart of the human is 2-chambered, 3-chambered or 4-chambered or single-chambered. You must have answered by now. Human beings are mammals and mammals have 4 chambers. Okay, that is 2 auricles and 2 ventricles, right and left auricles, right and left ventricles. Okay, so the answer of the question is third option, 4 chambers. Now moving to the next question. These questions are very simple, you can answer very easily. Which structure delays the impulse in the heart? That is AV node. Just before this we have discussed why it is delaying, because it requires time. Why it requires time? Because ventricles have to fill completely before contraction. If ventricles don't fill completely and they are contracting, what happens? Partially the blood enters the blood vessels. So AV nodes hold the impulse for some time. Okay, like if the impulse is transferred from SA node to AV node, it takes little more time, then the impulse is transferred to the bundle of His and Purkinje fiber. Okay, now moving to the next question. So here the answer is going to be AV node. AV node is the one which delays the impulse in the heart. Now coming to blood pressure is measured. The blood pressure is measured through mercury. Okay, mercury units. Okay, mm of Hg is the unit to measure blood pressure. And what is the blood pressure? 120 by 80. Normal average pressure is 120 by 80 mm of mercury. You must have seen blood pressure, old blood pressure machines in the clinics. We have bulb and we have a monitor where mercury is, whenever you pump the bulb, what happens? The mercury rises and falls. This is because we use mercury element for the measuring of the blood pressure. It is a liquid element which can be used in normal temperature for measuring blood pressure. Okay, so normal blood pressure 120, which is a systolic pressure, 80 is a diastolic pressure. Systolic, the amount of heart pressure is given on the contraction is called as, the blood has pressure while contraction is called as 120, and relaxation, the pressure required for relaxation is 80. Okay, so next, moving to the next question, normal systolic pressure in adult is 120 mm of Hg. Okay, what is diastolic pressure? 80 mm of Hg. So here the question is only about systolic pressure, so that is 120 mm of Hg. If the pressure is more, then it is high pressure. If the pressure is less, then it is low pressure or low BP we call it as. Now moving to the next question, which artery supplies blood to the kidneys? In the earlier session only we have discussed the kidney gets which artery? Renal artery. Whereas hepatic artery is supplying blood supply to the liver, coronary artery to the heart. Okay, heart also requires oxygenated blood, pulmonary artery for lungs, okay, hepatic for liver, and renal for kidney. The arc of aorta divides into multiple arteries, that is hepatic, renal artery, coronary artery, pulmonary artery, or artery that is supplying to the nervous tissue. Okay. Now here next question, the blood vessel that brings oxygenated blood to the left atrium is pulmonary vein. Now why we are calling it as vein? Vein doesn't carry oxygenated blood, right? Vein carries deoxygenated blood, but the definition of vein and artery is not like this. The vein carries blood towards the heart and the artery carries blood away from the heart. Although this is an exception over here, this vein is the only vein which carries oxygenated blood. Remaining all veins carry deoxygenated blood because it is carrying blood from lungs to the heart. Okay, so here the blood vessel that brings oxygenated blood to the left atrium is pulmonary vein. Don't get confused in this type of questions. They are very important NEET questions. So always understand the definition of pulmonary vein and pulmonary artery. Vein is the blood that is brought to towards the heart, and artery which means blood away from the heart. Okay, so that's how they got. Although it consists of oxygenated blood, but it is carrying towards the heart. That is the reason it is named as pulmonary vein. Now moving to the next question, our last question. Capillaries are more important because they pump blood. They don't pump blood. Blood is pumped by heart. Transfer of impulse that is done by pacemaker or like SA node, AV node or Purkinje fiber. Production of RBC that is done by our bone marrows, and exchange of substances. This is true. They are very single membrane, fine membrane structures which are helpful in exchange of substances. So this is our last question that we have discussed. Allow exchange of substances. So capillaries are present in the lungs also for exchange of gases. Capillaries are present in each and every structure so that they can exchange oxygen and carbon dioxide gases to different body tissue. Not only oxygen and gases, glucose or any other essential elements. Okay. So this is it. We have discussed about questions from cell and we have discussed questions from the heart structure, blood vessels. Okay. And SA node we have discussed about pacemaker also. Okay. Thank you so much. We have discussed worksheet 3 in botany as well as zoology.